Good morning, good afternoon, good evening, wherever you are. This is Mrs. Casper coming to you. This is for my private students. I'll be recording some lectures for you this year, so I'm looking forward to it. Thanks for getting a hold of me. I won't you be using your name to keep it private, but it's good to have you back in the fold. All right, so we're going to be covering Biology 1. Now, today is August of 2025, and I'm recording this lecture today, but some of the lectures you'll get will have been recorded previously, and many of those will have been recorded during an actual lab class, which presents a couple of problems. One, you will hear some background noise, you will hear some questions of students, maybe some jokes, some laughing, that kind of thing. I will try to go through those. If there are some that are just too much, then I will re-record those. Okay, but in order to save time, I have some of those already, recorded for you. Okay. The next thing is that we won't be doing any labs with this because this is a remote situation, but I understand some of my students will indeed have labs elsewhere, so that's great. I will give you ideas for labs as we go along. So that would be fun, and if you need to contact me about lab materials, I'd be interested in and happy to help with something along those lines. Okay, so here we are. I am, as you know, as some of you know, I am a biology teacher. I received my degree in microbiology from the University of Texas at Austin, Hook'em Horns. So biology is right in my wheelhouse, and I really, really enjoy teaching it. All right, we are going to be using the latest edition, the third edition of Apology of Biology. It's really great. It is quite different from the previous editions, mostly in the sense that it has different order of things. Okay, so we're going to go through this order because that's what most of the students will be using, would be the latest edition. And it looks like that. I don't think that as a private student you will have any lab notebook that you'll be using to record notes or to use for lab, write-ups or things like that. Perhaps you have that in your lab class. If you want to just take some notes along the way, I'll be giving you the major points in this module. Also, I will probably be sending to you the entire module. In other words, all of Module 1. Let me get to that schedule, which is right here. Okay, here's the general schedule. Generally, there are 16 modules in the book, and we cover usually them in 2 weeks' time. So here we have, today we'll be doing 1A, and I'll go ahead and send 1B because I have that one previously recorded. And notice we have it AB, AB, AB down here, and then we get down to just before Christmas. You may or may not be taking a midterm exam. I don't know. I'm not going to administer or grade it, so that is up to your folks if they want to do that. And then I wanted to show you here in the second part after Christmas. Notice here in Module 8, we'll only be spending 1 week on it. There's not really a lab or anything. We'll talk about the theory of evolution and how we can deal with that as Christians. And we don't ignore it. We don't have to ignore it. I'm not afraid of it. We can talk about it in a scientific manner, and it'll all work out great. So then we'll go through and start learning about some of these other things, right? Prokaryotes, and then the different kingdoms. And notice right here it says schedule change. That's because in Module 10 it's very long for one, but I also wanted to spend more time talking about fungi. Since I'm a microbiologist, this is right where I live. I like looking at things under the microscope. I'll be showing you microscopic slides as pictures on the lecture. If you have a microscope, then that would be great if you could look in that. Also, I think I did that again somewhere. Where did I do there? Here's AB. I feel like in 16 I might have an extra lecture right there. We'll have to wait till we get there to see if I did that. I just know we had to rush through class Mammalia and it made me crazy. So we'll see about that one. All right, so here we go. Biology. Module 1A, biology is the study of living things. That's what it means. Ology is to study and bio is life. So to study life is what we're looking at. Now, life would include all the things, not just animals, the things that we're kind of excited to learn about, but plants, fungi, mushrooms. Over here on the left, you can see this is a picture of some bacterium. Bacteria, of course, would be in a microbiology sense. That's the kind of thing that we would study. And we'll look very deeply into microbiology, into bacteria, and to see how bacteria are really our friend. There's nothing to be afraid of with bacteria. All right, I got a little video right here, I think. Oh no, I just have this. All right, so biology covers a plethora of things. Right here, we're going to see, look at all these different words on here. We have genomes, biology, respiration, carbohydrates, photosynthesis, genetics, enzymes, evolution's in there, genome. All these things are going to be part of the stuff we're learning about this year. Some of it can get a little bit tough, nothing too bad. I think probably cell division and cellular respiration is going to be the toughest thing, but mostly biology 2 covers that. All right, so how do we do science? All right, so one thing to know about science is it asks questions, okay? And we, through observation, try to answer the questions. And that questions get answered in respect to what data we can collect. It's not just an opinion, it's something we try to find through observation and through experimentation. All right, so we have something called the quantitative, which is an observation including numbers, counting, and measuring. In other words, we have a quantity there. It's usually fairly exact. If we're getting a liquid to use for a experiment, we might use it in milliliters or liters. We do use the metric system completely in science class, even here in America. But we also have something called the qualitative observations. They're not easily counted or measured, like color. In other words, we just look at it and say, well, it looks like it changed from orange to yellow. It looks like that. So maybe something happened in there. Things like that, texture, stuff like that. Okay, so we can have some exact things, that would be with the math, when the math comes in, and then some inexact things as well. All right, we also have something known as inference. This is to interpret the data that we have based on stuff we know, that's prior knowledge, based on experience, or based on evidence. Okay, so keep in mind that science can prove nothing. It can only disprove things. Okay, so science isn't about trying to prove things. It is about trying to find information, trying to answer questions. But we must always be ready for our answers to be questioned by somebody else and thrown out. Okay, so we can't hold on too tightly to what we think we know, quote unquote, scientifically. All right, but if we have some evidence from somebody else's experiments or something like that, right here, we can read about those things. We can take some background knowledge. Maybe we've interviewed a doctor or something like that. We get some more information, their opinion on something, and then we can infer from that. We can infer some things that might be true. But keep in mind that anything that's true in science can be proven untrue at some point. All right, a hypothesis. What is that? That's a suggested testable answer to a well-defined question, a scientific question, or a possible explanation for observations. Generally, if you have a hypothesis, it starts with I think. So I think this will happen when I do that. I will say that using the scientific method in the regular traditional sense by having a question and making a hypothesis and having an experiment to explain my hypothesis or to quote unquote prove or disprove my hypothesis is not as prevalently used in biology. Okay, so biology tends to be more about observation, and so our hypothesis would be something like, when I look at these cells under the microscope, I believe I will be more informed about these cells, something like that. We're not trying to set something up. Now, other times in biology, we might be looking, for instance, what is the best, fertilizer for a plant or something like that, and so our hypothesis would play into it that way. But keep in mind, probably 50-50 on doing experimental things that require a hypothesis and experimental things that just require observation. All right, what are variables? So variable is a part of the experiment that changes. All right, I think I have a definition here. It's a factor that changes in the experiment. We see a little girl here dropping some chemical down into a flask right there, and she's probably counting the drops and measuring how many drops it takes to change the color of the liquid, something along those lines. Okay, so then she would be keeping that data, writing that down. Here's a better look at it right here. So if we have, let's put this in real quick. Yeah, there we go. All right, so we have the variable that is being manipulated. Can you see that up there? Let me just see if I can move this up a little bit. There we go. All right. The thing that we're doing, so for instance, we have 3 plants in those pots down there. The same kind of plants, the same kind of pot. They're sitting in the same place in the lab or the house, in a windowsill or something. And what are we going to change? We're going to change the amount of water. This plant will have a moderate amount of water. This plant will have a lot of water, and this plant will have a little bit of water. Okay, so as time goes by and we watch our plants grow, all right, we will also have something known as a dependent variable that is affected by the change. So look what happened to these plants. So what's going on here? Check it out. What do you think happened to this poor middle plant right here? Remember, it had a lot of water. I believe we can conclude on this one that we gave that plant too much water. Did you know that plants can drown? They can. They can have too much water. And now this one is growing, but it's not growing really well. But look at this one. All right. So it looks to me like we could conclude from this experiment that this moderate amount of water is the right amount for this particular plant to grow in this particular way. This plant over here is growing, but maybe it could use a little more water. Obviously, this one had too much. All right. So here is a great, when you're trying to figure something out, you might use humans sometimes to figure these things out. All right, so for instance, right here we're looking at somebody who's going to try a new medication for blood pressure. So the patient here, that's the lady here on the left, she must be struggling with some high blood pressure. And so she comes in to be part of an experimental group to test a new medication. So here is the doctor or nurse who's going to be administering and then administering the medication and then taking blood pressure measurements to see if it's working or not. Okay, so but anytime we want to test something, we need to have something known as a control. All right, so you know, in our plant group back there, we could have put a plant in there that we did nothing to except gave it the same amount of water as, say, the medium one or something like that. We could have a control group there. So the control group does not have a manipulation. The reason with plants, they do have to have water, obviously. So, but the control group, in this particular case, this patient right here won't be getting the medicine at all. She'll be getting something called a placebo. And a placebo is a fake pill, or what we might call a sugar pill. All right? And then we have someone over here who's going to get the actual medicine that's being tested. That's the experimental group. That's the thing that gets manipulated. So why in the world do we want to do that? If you've taken general science, you learned about using this as a group to be tested. It turns out that psychology has a whole lot to do with how we respond to medication. If I give you a sugar pill and I tell you this pill will make you feel, you know, great, then because you believe me, it may make you feel great, but it was just a sugar pill. Okay, so we have to take that into consideration when we're looking at something. If a person thinks their medication will bring their blood pressure down, then maybe even a sugar pill will do that. I know that's hard to believe, but it is true. Okay, and so therefore we test against that. So now sometimes we'll know that our tester will know that we're getting a placebo. Sometimes the tester will not know who's getting the placebo. Somebody else behind the scenes knows. All right, so we get into what makes science science, and we'll be looking at things called scientific theory. All right, so if we want to have a scientific theory, what we're going to do is we're going to have a question. So we're going to try to explain something, all right? And we're going to try to test it through experiments. We see that right down here. An explanation of some part of the natural world that has been thoroughly tested with support from experiments, okay? And so what we're going to do is we're going to have a question. All right, and we're going to design an experiment to test. Now, what is the prediction? What word could we use here besides prediction? We could use the word hypothesis. Okay, so I have an idea or a question. I make a prediction or a hypothesis. I design an experiment to test it. I do that experiment. I collect the data, observing it. Then I either say, yes, my theory was correct, or no, my theory was wrong. And it's okay to have a wrong theory as long as you say that, you know. And so, but it's just the way to get to something. So we have a lot of scientific theories, okay? These are things that have been tested and tested and tested over time, and they've held up so far, but they can always be overturned. All right, then it gets to what is known as a scientific law. All right, for something to be considered a scientific law, the test has to be done over and over and over again. All right, so it's a description of a natural relationship or principle often expressed in mathematical terms. I'll bring that in right here. Look at that math, yikes. to support the evidence. Okay, I'm going to go back for a second. This is a picture here of Isaac Newton. Notice he's looking at an apple on the ground. It fell from this apple tree. Therefore, he's going to start making conclusions or asking questions of why does the apple fall down all the time. It's not that Isaac Newton invented the theory of gravity. What he did was he applied the theory of gravity to it's the same force that's, although some people would get upset with me calling it a force, okay, but it is the same thing that is holding the moon to go around the earth and the earth to go around the sun. In other words, he came up with the scientific law of universal gravity. All right, and he did indeed use math. In fact, he had to invent calculus. So thank you, Isaac Newton, for inventing calculus. Yikes. All right, so what's the law versus a theory? Okay, so the law is something, like I said, that's been tested and tested and tested for a long, long, long time. Nobody's been able to overturn it. I'm talking hundreds of years. Okay, versus a theory. Okay, the theory says, I think this is happening. I think this is why it's happening, but I know that I may be wrong. Okay, so every good scientist would be able to say, and should be humble enough to say, I may be wrong. All right, so does a scientific law always stay a law? Does that mean it's for sure 100% of the time? Nope, it's not. All right, so if we have a theory why we have natural things going on, that's my idea, my guess really of what it is. The law summarizes that's been happening for a long, long time. They're things that are true for both of them. We have based on a hypothesis, can be used to make prediction, and can be revised. Notice can be revised is in the middle, meaning I can revise a theory, throw it out. I can revise a law and throw it out. It's a famous one of those. All right, so what do we need? Here it says on page 9 in the textbook, it's going to tell you how we do this. Okay, so we have a question. We make a guess. That's our hypothesis. I think this is going to happen. I either do an experiment or design an experiment. I write down my data, my results. I draw conclusions, and it's either a supported hypothesis, I was right, or not supported, I was wrong. And then you write your conclusions as a reported result to say where you think maybe the experiment went wrong, what you could do better next time, or if your experiment went and supported your hypothesis, you'll make a little conclusion there. Okay, so we have a conclusion of results, a conclusion of data, and we have a conclusion of the experiment. If you're writing up a nice experiment for a teacher or for somebody in another place, your parents in another place, be sure to always write a nice little conclusion at the end telling me what you learned doing that experiment. All right, so here are the steps of the scientific method. This is something that students who are taking this class and being tested would have to know, and they'd have to write these down on a quiz and possibly on the midterm and even possibly on the final exam, because it's something that's that important that would go for the whole year. All right, so number 1, make observations, form a question. In fact, if you're just curious about something. I love it when students, like for instance, I had a student one time who was very interested in bread, baking bread. And so she had a question. Well, do I have to use that amount of yeast, or can I use a different amount of yeast? I said, do an experiment. So in her question was, how much yeast will make a good loaf of bread? So she used one with the recipe, then she used one with less yeast, then she used one with more yeast, and then she let those rise, and then they ate them. They ate the bread, and then they decided which one was the fluffiest or the best or whatever. And you can learn things that way. Sometimes you personally might like a fluffier bread than somebody who likes a more dense bread, something like that. So that person had designed an experiment to test that question and that hypothesis. It is important to make a prediction beforehand to say, I think that the recipe will have the best bread, but like I said, you might have had a fluffier bread with more yeast, so who knows? You analyze the data, you draw conclusions, and then you determine whether you were correct or not in your hypothesis. If you need to do it over again, make some more bread, go for it. All right, limitations of science. I've been hinting at this the whole time. It's very important for you to remember the things that science cannot do. I love the cartoon over here on the right. It says here, and this new discovery completely changes everything you were ever taught about the origin of life. Oh, wait. A newer discovery totally changed what I just reported. Okay, I don't know if you've heard very, very recently, like this summer, the summer of 2025, all of a sudden scientists are saying, I don't think the Big Bang Theory is really one that's supported anymore. Okay, so for years they've told us that, and now they're saying, nope, it's not good anymore. All right, whatever. I don't know how much data we can collect on the beginning of the universe. That's a hard one to study because we can't go back in time and watch it or anything like that. Okay, but just keep in mind, It's okay to question science. It's absolutely mandatory to question science. Never listen to someone wearing a white coat saying, science has proven, and they go on, just forget it. They don't know what they're talking about. Science has proven nothing. Okay, so what are we going to write down here? That science cannot prove anything. Okay, we always think it's science, great. Oh no, science has its limitations. Here we go. Science cannot answer all questions, but we can certainly ask them, and we can certainly try. All right, bias is can be a factor. What it means is a person who prefers an idea and possibly does not give equal chance to a different idea. In other words, if I'm going to do a study on the origins of the world and I'm already a creation scientist believer, in other words, I believe God did it, I'm going to lean toward the data that shows me that. I will say, and when we get to Module 8, we'll talk about all these things. I will say that there's plenty of evidence to show creation, or at least design, right? I believe in creation, of course. I also believe in design because I believe God is a great designer. All right, now, there was a law for some time, or a theory, and one of them was called the spontaneous generation theory. So look at that word. Spontaneous means happening naturally. Generation, being created. Okay, you're looking at a really gross picture over there. Sorry about that, but that's how this experiment was studied. Way back in the day, we're going to see how people thought that flies came from rotting meat. Okay, so this is a theory that living things originate from non-living things. All right, this is a picture, somebody's idea of what Aristotle may have looked like. Aristotle was a Greek thinker, a Greek teacher. He was actually the teacher of Alexander the Great. He was a brilliant man. Okay, so don't get that wrong. He is absolutely brilliant, one of the smartest people ever to have walked on the earth. He was before Christ, of course, and he was not in the Jewish faith, so he did not have a real firm understanding of God, but he did have really excellent powers of observation. So he was able to look at things and ask questions and devise experiments. He actually also began naming living things. That's kind of the antiquated version of that. But one of the things he stated was that he believed that flies came from rotting meat, because you put rotting meat out, you put meat out in the sun, and after a while some maggots start to climb out of it and flies. That's the baby fly, right? He also believed if you left a bowl of wheat berries in a closet with a kind of a cloth tucked on top of it, that mice would sprout from that. Okay, but he was missing something, wasn't he? He was missing a variable that he had not prepared for. And in the case of the meat, the variable was that flies were landing on the meat and laying their eggs. In the case of the mice and the wheat berries, the mice were digging through the closet and finding the wheat berries to eat them, and that's how they got there. Okay, so for hundreds of years, the idea of Aristotle of the spontaneous generation held, because he was such a revered and respected scientist of old. Nobody wanted to question him. But as time progressed, he was brilliant, but he was wrong. As time progressed, scientists finally got brave enough to question it. They knew that there was no way that that was true. Now this is a picture, a cartoon picture, we're going to watch a video in just a second, of a guy called Francesco Redi. And Redi did an experiment, and this experiment is going to be taken to another level here in a minute by one of my heroes. And he put the rotting meat into jars. Now one of the jars here in the middle, he put a cork on the top so no flies could get down in there. But people criticized this because they said, well, if there's no air, maybe they can't grow because there's no air, but there, of course, was air in there. So he did this one right here by putting the mesh over the top, letting air get down in here, and then leaving one open. Now, this is a little bit not quite exactly how the experiment went. Certainly, we did have flies coming out of the open one, but we also had, over time, flies coming out of this, because sometimes some of those eggs, they did lay their eggs on top, and they did drop down in there, okay? But let's watch a little video on it.
Francesco Redi was an Italian scientist. His investigation was one of the earliest examples of using controls to test hypotheses. Let's see how Redi carried out the investigation. Redi left a piece of fresh meat on a table. After a few days, Redi found that some flies moved around the meat and some maggots appeared on the meat. question came to Reddy's mind. Why are maggots found on rotting meat? Reddy suggested a reasonable answer to his question. This answer, which tries to explain what is observed, is called a hypothesis. My hypothesis is the flies produce the maggots. Reddy predicted that maggots would not appear if he kept the meat away from the flies. Reddy tested the hypothesis by doing experiments. He prepared 2 jars and put a piece of fresh meat in each of them. One of the jars was not covered. It was called the experimental setup. The other jar was covered. It was called the control setup. After a few days, maggots appeared in the uncovered jar, and no maggots were found in the covered jar. Reddy found that the experimental results supported his hypothesis. He made a conclusion based on the results. I conclude that the maggots are produced by the flies.
All right, there we go. Okay. It's a little bit silly. Now, like I said earlier, we did have a sealed flask. I think he did more than one experiment is why we have it. We had the sealed flask, we have the gauze-covered flask, and then we have the open flask. And furthermore, another thing about scientific method is if you write everything down, really well, other scientists can come along behind you and repeat it. So it is important for a scientific theory to be formed. It has to be an experiment that's repeatable, that someone else can take your data, they can take your experimental setup and reproduce it and have the same exact results. Which is another reason why it's so important to write up your labs very thoroughly, because other people need to be able to repeat those. There have been some people in the past who have made these outlandish claims about, cloning humans and stuff like this, and other scientists say, oh, well, I'm very interested, show me your data. And yeah, they don't really have it. So it doesn't quite work out because they didn't. In other words, scientists will also fib a little here and there in order to get famous or to get people to support their research. In other words, give them money. Okay, so always be dubious, always ask questions. All right, so this lab would have included understanding how to be safe in laboratory times, okay, because in my regular setup, I, of course, would have been doing labs. So if you're doing labs elsewhere, then this might be useful for you. Really quickly, we'll go through this. Going to lab, we have safety contracts. That's what One Day Academy used to have. Listen to your instructions. Don't goof around. And in fact, you may have some labs to do at home, and just keep in mind to be safe about those, of spilling things and stuff. Goggles are a good idea, especially when working with chemicals, liquid. Appropriate clothing, that would be, you don't want your sleeve falling down into some chemicals, or if you happen to be using a flame, you don't want your sleeve of your, dress or whatever falling down into the flame. And then, of course, never remove any lab supplies from the place where they're being used, the kitchen, etc. So here are the things we use during lab time. Goggles, I'm going to show you a little video here about why goggles are important.
Egg whites are mostly proteins, similar to the proteins found in your eyes. In this demonstration, we have added an egg white to a petri dish. What we're going to do now is we're going to add some acid to the egg whites to simulate a splash in your eyes. Almost instantly, the proteins are denatured. This is also what occurs when eggs are cooked. There is no reversing this damage. Even when I add some base, the reaction is not reversed. This damage is permanent. Please, wear your safety goggles whenever working with hazardous chemicals.
All right, there you go. Chemical spills, in the case of working with science glass or even in your kitchen or whatever, broken glass is one thing, and then chemicals, even vinegar. One time at one of the schools I was working, it was a church, we spilled alcohol, just rubbing alcohol, but it spilled on the floor and I didn't know it. It's not really a problem. There's nothing wrong with rubbing alcohol, except when it gets to the waxy cover of a floor, and it ruined the waxy cover of the floor, and I had to pay for the floor to get fixed. Okay, there is also, if you're in a laboratory in a university setting, you will have something known as eye wash. Okay, and the eye wash looks like this. Here we can see somebody washing their eyes, and this real thing, the guy was screaming like crazy because, that gets chemicals out of your eyes. Here we go, wafting. I'm gonna play this real quick. If you're gonna smell anything in science, always do it this way instead of just sticking your nose right up to it. Waft it towards you. All right, in case of fire, you probably have fire safety instruction at your own home. Stop, drop, and roll. And then in biology, you will need to know how to be careful with a scalpel because you can cut yourself. The scalpels we use to dissect biological specimens are the same scalpels surgeons use in the surgery room, okay? So we don't want to have to use any Band-Aids around here. All right. Microscope, hopefully you have microscope available to you. I did mention that when we talk about microscopic samples, I will show you some of those pictures on this presentation, so you'll see some of those that way. But microscope safety would just be to really care for the microscope because they're expensive pieces of equipment. You don't want to drop them. Don't hand them around to other people or anything like that. Always make sure to treat them with great respect. And then just wipe off the where your eyes are looking in for the next student. All right, if there's an accident, just notify your parent or your teacher. Don't panic, follow instructions, and clean up at the end. All right, and I think that we're at the end here, safety first. So here we go. That's part 1 of module 1. It's a very general idea of what biology is. So I hope that was helpful to you. I will be sending part 2 along with this one as well. All right, I hope you enjoyed that, and I will see you or hear you next time. All righty, so long.